Awesome. Well, thanks to Simon and Khaled and all the other organizers of what is going to be a legendary seminar series on biosensors, I'm sure. Um, Really excited to be, one of the early speakers on this, and I'm excited to see what this comes up. So, what I'm going to do, I'm actually going to share a talk that I gave a couple of weeks ago at the Cell-Free Synthetic Biology Conference that was held at Northwestern. It was a great meeting of the field, and as you might imagine, biosensing was one of the major topics of discussion. And it was a really interesting time for me to actually take a step back and reflect on our work in this space and kind of where the field has gone. So I'm going to kick this off. Um, yeah, so what I wanted to do is, in the few short minutes that I have, is give a perspective of at least where I think biosensors research has gone and where does it need to go for it to achieve the impact that I think many of us, especially on this group, want to see it happen. I'm going to kick it off with a little bit of a historical perspective. This is a photograph of the 2015 Cold Spring Harbor Laboratory Synthetic Biology summer course. This is a course that was instrumental in creating at the very beginning, and I was lucky enough, along with Dave Savage, Carmela Haynes, and Jeff Tabor, to be some of the first creators and instructors of this course. 2015 was the third year of the course that I taught. And in 2014, I had seen a talk by Jim Collins that talked about his work with Keith Pardee in creating paper-based gene networks. And I think many of us on the call probably saw this, but it really showed the power of what you can do with lyophilized cell-free systems. And I immediately thought, wow, biosensing would be a really cool application space. Keith and Jim had shown some told switch stuff, but what if you could expand it to all the different chemicals that you might find interesting to be able to sense? And so, not really knowing how to do this from a research perspective, I took it to Cold Spring Harbor as our little play project at that year and played around, put a biosensor in a cell-free system, see if it works. Way in the back of that photo, I don't know if you can see it, is Khaled, who was one of the students of that course, who's still a PhD student at the time. And I don't know what happened, but some ideas triggered in his mind, and eventually Khaled joined my lab as a postdoc and was really the leader in getting us into this space right at the same time we were moving to Northwestern University. So it's been about 10 plus years since we got into this, and it's a nice time to take stock and celebrate all the amazing things that the field has done, and then think about what we need to do for over the next 10 years and hopefully shorter to achieve impact. Um, I'm going to personally thank many amazing people that have been through the lab. This is not everyone. These are people whose work I'll show today. Huge list of collaborators that I'll try to highlight the best I can, and of course, funding agencies. But this is really not just a large collaborative effort. This is really the effort of the whole field that I'm going to try to highlight. I'll start off by really kind of showing what we think is the promise of this. And sorry, I'm trying to hide my meeting controls. There's basically a huge potential for cell-free biosensors in the world that we can all recognize. There's many different application spaces for this technology. But I'll just give you some numbers on one specific one that we've been particularly interested in the water quality space. So this is a map, a little bit of an older map at this point, of essentially one aspect of something called water insecurity, which is, people not having access to enough clean water for their daily uses. And if you kind of go through the map, you would realize that about 2 billion people, or roughly a quarter of the people on the planet, don't have enough clean water to drink or for their daily uses. And a big part of this is knowing which water supplies are clean or not. And so you can imagine some sort of sensing technology that can measure water contaminants could have huge global impact. There's also quite a sizable market for this. These are numbers that Khaled crunched. So at about $11 billion market in global water monitoring alone, with a pretty impressive growth rate. And so that's great. There's a huge potential for technologies that can measure water quality. The problem is, as we all realize, the state-of-the-art technologies are usually laboratory instruments that have a lot of infrastructure requirement, a lot of cost, a lot of expertise, and a lot of logistics, because oftentimes these things have to sit in a room, so you need to collect samples and get to them. And that really doesn't scale to certainly the at the 2 billion person scale of these sensing technologies. What we'd all like to do is to transition to something like this. You've got a freeze-dried, cell-free biosensing system that you can rehydrate with the water sample of interest and see some sort of detectable signal. And it's so simple that it can get into the hands of real users to really meet the scaling. So that's kind of what the potential for this technology is. It's to enable anyone, anywhere to measure something important to them, be it water quality or something else. So the big question in my mind, as I reflect on what we've done in this field and others, is what will it really take for global adoption of this technology? I think we've made an amazing amount of progress on the technology side of things. There's still some really good, interesting challenges to think about, but there's some also big framing questions that we need to answer. So that's what I want to do. Kind of go through, where have we been over the last, X number of years here. We've got 4,500 publications in this space. We've done a lot of great work. Where have we gone, and then where we need to go? So where are we going? I'm going to do this relatively quickly. I think many people are probably already familiar with this, but I'm going to do my highlight reel, thinking about the platforms of this technology, some of the sensor work, the circuits, materials, and devices now that are being enabled. I will apologize in advance if I don't feature your favorite piece of work or your work, then I'm sorry. But I will point you to a really nice review that Brad Bundy recently pointed out that is more comprehensive than I can be here. And there's a citation right there. Okay, so what have we done in platforms? Early work in cell-free biosensors used bacterial extract systems. These are basically ground up cells. You get all the cellular machinery out of them. You supplement them with energy sources and cofactors and whatnot. They have a lot of really great advantages. They have a lot of machinery. They can do not only transcription, but translation. They can do native transcription, more complex regulatory methods. You can add lipids in these things, all sorts of stuff. You can scale their synthesis. You can grow tens, hundreds, thousands of liters of bacteria and make extract with them. Methods are improving there. And they have some nice advantages in terms of shelf stability, in terms of how long they stay on the shelf after you lyo them and still function. Major disadvantages, though, is their reproducibility. Still a lot of batch-to-batch variability of this. And so thinking about making products that make it out into the world, this is more of a challenging system to deal with. Partly to address that, and for other reasons, we worked on more purified systems. This is this Rosalind platform that Khaled was the major driver of when he was in the lab, named in homage to Rosalind Franklin. These are basically purified systems, so that's nice. They're defined. They're really tunable because you can change the concentration of all the different components, and you can adapt them to existing supply chains that make all these components already. Disadvantages is they've got a little bit limited machinery. The ones that we developed, they use T7 RNA polymerase, but not everything is compatible with T7. They do have a higher cost, and shelf stability remains a challenge. We are starting to address some of these. So Anibal Arce, who's a postdoc in my group now, is developing what he calls the eRosalind platform, which basically gets a much more sophisticated E. coli transcription factor system in here so that you can support other biosensing mechanisms. There's progress there. What about sensors? So up on the upper left, probably most work has been done on these APO repressor ATFs. Think something like TET-R, which binds to an operator sequence and blocks transcription, but then releases when the ligand is bound and creates a signal. This is work that Kirsten Jung and Khaled did in the Rosalind platform, and these things can sense all sorts of stuff: antibiotics, disinfectants, plant alkaloids, you name it. So that's really great work in that space. There's also a lot of really cool work expanding transcription factors. So in the middle left there, that's something out of Mike Jewett's lab, showing how these MER-R family transcription factors can be gotten to work in cell-free systems. It's a whole different class of transcription factors, and they're really good at sensing metals and other things, but that's really exciting when you expand it to a new class. And then some work in my lab and others, Ram Naji here, expanding into these LTTR ATFs, which are more complicated, but they're one of the largest families of transcription factors known. So that would be a huge expansion of what we could sense if we can get a hold on those. There's also other great sensors. On the upper right, we've had some really great success with RNA riboswitches to sense things like fluoride, which is at high levels a toxic compound that you want to detect in water. Um, in the right middle, there's really great work out of Rebecca Schulman's lab showing how you can graft aptamers directly into a transcription template and have them regulate T7 RNAP transcription. And so they've been gotten a lot of mileage out of sensing proteins with that. They call it the ARTISTS platform. And on the bottom right is something that just came out of Mark Stasinski's lab at Georgia Tech, using a split T7 system fused to nanobodies as another method of sensing proteins. So a lot of great work in sensors. Of course, we always need more sensors, but as a field, we have a pretty good handle on these systems. I think the last thing I'll say about sensors is, of course, the elephant in the room, which is how is AI going to help this? And I think the early clues is going to help us a lot. This is work coming out of Mike Jewett's lab with a pretty clever tweak on a machine learning algorithm that they apply to change the specificity of a metal sensing transcription factor, which is something that chemically is extremely hard to do, but they have found success. That bodes well for the future. What about circuits? Well, when cells use biosensors, I think one of the coolest things is they often don't just do something with the signal directly. Instead, that signal goes through a genetic circuit, which can then process that information and help the cell do something with it. There's been a lot of cool work on incorporating circuits, and I would say probably the coolest one in my mind, and I know this is maybe tooting my own horn here, but is recent work that Kirsten and Khaled did also with Jenny Lee, who recently graduated from the lab, using what's called toehold mediated strand displacement circuits to process information from biosensors. TMSD circuits for short are really cool technologies straight out of nanotech. It's the foundation for DNA computing. And essentially what you can think of it is a highly programmable genetic circuit architecture where you're using DNA base pairing and the exchange of strands between these nucleic acid gates and free strands to process information. The DNA nanocomputing people have figured out how to do amazing things with this platform, and our trick was to figure out how to make it compatible with the biosensing mechanisms. We can do stuff like logic gates and out the wazoo, and that's interesting. They might find use in applications as more proof of concept. Even cooler is we can design circuits that can control the timing of biosensor signals. So you can control the kinetics of TMSD gates very easily. And when you do, you can imagine creating different thresholds of sensitivity. So in this test format, people were asking, can I get more than just a yes, no answer on whether my contaminant is present? And so what we did is we create a series of tests. They all sense the same thing, but at a different threshold. And so you basically count how many tubes light up to get an idea of a semi-quantitation of that. And then the latest circuits we've developed is actually a really cool trick of T7 transcription, where it will do some off-target transcription, and you can use this to regenerate signals. We call this polymerase strand recycling. And when you have this amplification system, it turns out that you can actually improve the sensitivity of sensors and shift these dose response curves to the left. So a lot of great stuff in circuits. Probably going forward, I think an exciting area for circuits is what's called these co-transcriptional strand displacement gates that are coming out of Sam Schafter's group at NIST, where they don't have to encode these gates in DNA, which can be a little bit tricky to prepare, but they do it in an RNA molecule that they can then transcribe, which has a potential to scale this circuit platform. Been a lot of really cool interfaces with materials as well, not just cell-free sensors and a tube. This is work that we did with Neha Kamath's group at Northwestern, where we encapsulated here the fluoride riboswitch sensor in a membrane and showed that the system confers some protection against other things in the environment that might interfere with its function. And then Dylan Brown in the lab has been recently playing with other types of material additives. Here's melanin nanoparticles which can be added to cell-free reactions to offer some protection, for example, to UV light exposure. And then there's a lot of great work in devices. So upper left, this is out of Mark Stasinski's lab showing you can use enzymes to make colorimetric outputs for easy visualization. This work was also really cool because they came up with a calibration scheme to control for matrix effects. If you have some inhibition in your sample, you can control for that. Um, Fluorescence is obviously really important. This is the handheld 3D printed handheld illuminator that Khaled made, which is really cool to be able to take roslin out into the field since it uses an RNA output to generate fluorescence. Diana Capdevia's lab on the bottom left has just came out with a preprint that kind of uses the same roslin platform, but it has a lateral flow strip assay readout, which is really exciting because these are things that people use all the time, for example, on COVID tests. And then there's some really cool work, a little bit still nascent, but trying to get electronic signals out of biosensors. So on the left and this bottom right panel is work out of Keith Pardee's lab that he did with Shana Kelly, who's now at Northwestern, basically using the cell-free system to make an enzyme that can make a redox reaction with a circuit. And then work we've done on the bottom right using a nanomechanical cantilever system that, you basically get the transcription factor to cause deflection in a cantilever and generate a signal. And electronic interfaces are really cool because then you can do a lot of stuff with that information, post-processing, collection, communication. Question is, so we've done a lot of stuff and done a lot of cool tricks, but where do we need to go to get real impact here? This is a photo of the Cell Free conference that I mentioned, where had some of the best people in the world thinking through this. Well, I think a major first step is we need to get into the field more. These technologies work great in the lab, but as with everything, you put it out in the field, you'll learn when they work and when they don't and how to improve them. These are a couple of really cool articles that Keith Pardee's lab has been doing. I think they're doing some of the best work in this space, either getting medical diagnostics out into different parts of the world or thinking about local manufacturing. So these are two things that I think would be worth following up on. Then I'd like to share a little bit of our work on trying to do this and our path and what we've learned, which is a lot of really interesting things. In 2018, this is our first chance to do this. We had developed a fluoride riboswitch biosensor, and it worked great in the lab. And this is a photo of Matt Verisloff, who selflessly volunteered to go to Costa Rica to try to test this thing in the field. Why Costa Rica? Well, we didn't really know anything what we're doing field testing, and so we looked up where in the world were there high deposits of fluoride, and we found a poster online from a dental conference that said there's dental fluorosis near the Irazu volcano in Costa Rica. Turns out anytime you're near a volcano, you probably have a lot of fluoride. And so Matt took out a few tests. We really didn't know what we were doing, but excitingly, the controls worked, and he did find some field samples that showed signal, which is, to us, this was really great. The lesson was, you can take these things out into the field and have them work. Obviously, we needed a lot more data, though. So that was the major improvement that we needed. So to try to get this in 2019, we took Rosalind out to California. Khaled, Kirsten, and I and our collaborators went out there to test for metals in the groundwater because the wildfire in Paradise had melted all those municipal structures. This is really important, actually. With our first trip out there, many of these tests failed. Probably half of them failed, which we were able to trace down to a packaging issue. We weren't as rigorous in our packaging as we should have been, but once we improved that, we showed that we were able to manufacture tests at Northwestern, ship them out to California, and they worked great in our collaborators' hands. So that was an improvement, but I think a critical thing was the desire to get non-expert data. It's one thing for the people that develop the test to go out and use them in the field, but what happens when you get them in the hands of someone that hasn't seen this before? And those are largely the people that we want to help the most. Well, to do this, we needed to expand the team. And we included two anthropologists. Sarah Young is an anthropologist here at Northwestern that does a lot of the global health water space. And Patrick Uhr, at the time, was her PhD student. Now he's a professor at Wayne State University. And we went back to Kenya with the fluoride test. And we went to this place called Nakuru Valley, which has a lot of fluoride. and we gave the test to real, household members, um, and asked them to use it. And the great news was that they could use this test, even though it was very kind of crude first version of this, so not a lot of optimization on packaging or user interface, but they could use it. The test performed pretty well, decent numbers on sensitivity and specificity, the diagnostic calculations of those. And also, I think the really great thing is that there was early indications that these tests were not only usable, but they were effective. And effective meaning that the ability to do the test would change people's knowledge, attitudes, and behaviors towards water quality. For example, in this part of the world, there's two places, major places where you can get your water. One is through a borehole, which often has fluoride contamination, and one is through rainwater collection. And we could give tests to people and they could test both and see for themselves that the borehole, that test lit up, but the rainwater didn't. And so then they would know when they can to get the rainwater and use that. So this is a really, great indication that there is something there behind this technology. We did find some technical things like shelf stability was really tricky in a place as hot as Kenya. So that's something that we need to improve on as a field. We've been doing this more and more and more. So this is Herma Demisi, who has been working at a Gates Foundation sponsored project actually to do crop pathogen detection. I put the map of the collaborators on this because it's an absolute global team. Here, the idea that Gates Foundation thought they could empower farmers with diagnostics to see if their crops had pathogens in them and then do something about it. We're going in with essentially a version of the Sherlock technology that uses isothermal amplification plus CRISPR-Cas13 with some interesting tweaks, but that's largely the theme. We've taken this out into the field. The test isn't as far along as some of the other ones, so we're bringing out much more equipment than we'd ever want to. But this is some photos from the field. And the upper right, this is us celebrating our first positive hit literally at the equator, which is kind of fun. But we've learned a ton about this. One, we've learned about real logistical challenges when working in parts of the world like this. Test stability is a major issue. There's a photo of me in a field that I am basically melting. And if I was melting, the test was melting. But probably the most important thing is on the right, bottom right, which is this ecosystem map. What we realized is that even while Gates Foundation had a vision for farmers being able to use this technology, we quickly realized that farmers are probably not the ideal stakeholder for this, both because of some of the extra steps that the test needs to be done, but also they're not the ones that might be able to really take action on the information provided by the test. And so we thought that we started to do our own stakeholder maps, but really our thinking changed and realizing we need to be working with stakeholders as soon as possible from the very beginning of test development. And that's what we've done in our latest field trial, which is an ongoing field trial in our own backyard in the city of Chicago to detect lead in drinking water. Chicago has the highest number of lead pipes in the country, if not the world. It's a big, big problem. And we wanted to help address this problem. Again, to do this, we had to expand the team. So we're involving Sarah and her team as well. But now we've got Rob Weinstock of the Northwestern Environmental Law Clinic. We've got civil engineers that are experts in water chemistry. And we even have community members. Vanessa Bly on the bottom right is the leader of our field team that goes into people's homes and gives them these tests to work with. And if you're interested, this website that we've set up has a lot more information about this study, which we recently completed round 2 of, and we're about to launch round 3 in about a week. We also incorporated stakeholders through creating a community advisory board. These are local politicians, local people that live in the areas, environmental advocates, people that are familiar with water issues, and we partner with them to try to understand how do we communicate what we're doing to people, how do we get them enrolled in our studies, how do we communicate results from the study. We even collaborated with a local artist to make a comic book that is accessible on the website for people to get at and figure out, understand what we're doing. We're doing something that you might imagine is pretty similar, where we have these biosensor tests. We meet with the community advisory board to understand how to interface with the community, recruit participants. We give people tests in their home, and I'll explain more about this later, but then we get the results, we compare, and we improve the tests. The test kit currently looks like this. It's a lateral flow assay. It's done in about 25 minutes. So you put a drop of water in, you wait about 10 minutes, you put a couple more drops of running buffer in, you put your strip in, you wait 15 minutes, and then you look at it, compare it to a scale to see how much lead you have. We also give them social science instruments to measure, for example, what is their knowledge about lead? What are their attitudes and behaviors about it? So we can do this before the test and after the test to understand how people's behaviors are changing. And of course, we give them all sorts of instructions and filters and whatnot to do, things to do if we find a positive hit. I won't present any data from the study since it is ongoing, but these are some of the field photos from the field showing the variety of different people that are using the test, all the way from, a mother and a small child to people that are quite old that are really want to know if their water is clean to drink. And I will say anecdotally, the results are astounding. The test is working extremely well, and there's huge desire for this technology out there. So that's really exciting. Look for more soon. Um, Finally, I'm just going to end on some 5 questions that I think we all need to be asking ourselves as we develop these technologies to really try to make them as impactful as they can be. In addition to just doing more field testing, which we all need to do, these are questions that really frame what we're trying to do in the first place. You might think that they're questions that are most appropriate for companies and things like that, and that's certainly true. But I think these questions are broad enough, and there's a lot of work that we can do even as an academic community to ease that path towards broader adoption in the commercial space. So I put Ashti Karim's photo. Ashti's a assistant professor here in the Center for Synthetic Biology. He and I are developing a SynBio textbook together with a lot of other people, and these are some of the ways that we're trying to frame some of what's going to be in that textbook. Okay, first question, what are the markets? There's a lot of different application spaces. I talk mostly about water. That's what we're passionate in, but I mentioned ag. You can imagine human health. There's things with food quality. There's even thoughts that these biosensors might be enabling for space. And so the question is, what markets are we really working towards? Because the ultimate, look and feel and version of that technology is going to depend on these choices. Understanding who will pay, what are the business models, those are all really important. And the tools are something that academics don't do a ton of, but we can do more of, like customer discovery and market analysis and all sorts of business stuff. The other question is, are there any existing regulatory frameworks that are important to consider with these technologies? A lot of different, it kind of depends on the market. For example, if you're looking at a drinking water thing, maybe you're looking at an EPA regulatory framework. Obviously, if you're in human health, you're looking at an FDA. Are there things like export controls and tariffs, things that we've accidentally stumbled upon when we try to do our Kenya work, where we had to get clearance to even take the technology out there for field evaluation? And then are there safety frameworks? I don't imagine there's a lot that's too big of a concern, but we should really map those out. Really important question, how do we scale? So, can we leverage existing infrastructure? On the top here is an image from a paper by Keith Pardee, which I think is extremely clever, basically trying to get cell-free biosensors to work with a glucose meter. Why is that important? Well, the glucose meter is probably one of the most widely distributed and used diagnostic handheld devices in the world. So arguably, the distribution development of that platform is already done. If we can get our test to work on it, we are almost home free. So that's a really great clever thing. Can we do more of that? On the bottom is an LFA manufacturing platform. LFAs are great because there are scalable things like that. There's also really great work by, for example, Jenny Malloy in Cambridge and others to think about local production of materials. Sometimes logistics is really a barrier for low and middle income countries. And so local production might be important, but is it even allowable and is it doable? Are there IP restrictions that prohibit scaling? Again, are tariffs and export controls going to inhibit scaling? And I think these things are really important because I need to know, am I developing an LFA or if I'm developing towards a different device? And I should know that from the very beginning. Big question, is there a role for policy? Policy can be inhibitory, but can also be enabling. This is an interesting anecdote. Um, the lead, there's something called lead and copper rule, which in the US regulates how much lead can be in drinking water. And it was recently redone. And when it went under a revision, it had a public commentary period. And so Rob Weinstock helped us write a question or a comment to ask the EPA to consider the use of cell-free biosensors in monitoring lead for drinking water. Now, the EPA has a very specific definition of what a certified method is that can be used to satisfy the LCR. And it's basically a laboratory method because they focus on reproducibility and so on and so forth. So they were not willing to necessarily replace that method with a cell-free biosensor thing. But the great thing about public commentary is that they have to respond to it, and they did respond. And in their response, they say qualitative at-home tests are useful for assessing the potential presence of lead in drinking water, but not for making quantitative assessments. So I think this is critical because they are acknowledging there's a potential for this technology. And they're also pointing to a limitation. And what their limitation here, they say it's not quantitative because it's not known if the tests get at total lead or dissolved lead. But if we can develop a version that gets at total lead, maybe you could go back to the EPA and get them to say, hey, this is now an official method that governments can use to satisfy these legal requirements. And if you have that, then this policy goes from being inhibitory to enabling, showing people that they can trust this technology. So I think there's a lot of other types of policy dimensions with this, especially for LMICs, low and middle income countries. And we just, as an academic community, need to have a better, more sophisticated understanding of this. And then finally, what are the technological challenges that need to be solved? This is the stuff that we already know and love. We do a lot of this great stuff, and basically, if we answer those first four questions, we'll be pointing the ship in the right direction as we do this. Okay, I spoke for too long. The final thing I want to say is we need to get the word out in general. This is something we can all do as a community. I showed you what we're doing in the case of our Chicago lead study with our comic book. I'm showing you some other news items. It turns WBEZ Chicago has been embedding a reporter in our field study and help us communicate the importance of our work this way. This is Tyler Lucci, one of the key developers focused on CBS Chicago, came to the lab and filmed. There's also, some unfortunate things happening with just public mistrust of science and losing support in the public eye. And when Northwestern got handed down its funding cuts of $790 million a few months ago, this gave us an opportunity to go to the public and say, what does this mean when you cut this? And in this case, it means cutting some of this work to make water quality monitoring accessible to everyone, which is something that benefits everybody. And I will say it's very effective. Having a sensing technology that tells people if they're healthy or not is something that everybody can get their head around. And so I encourage you to try to do more of that out into the world.